Hi everybody, this is David Lally at the Partnership for Research and Education in Plants, and today I'm going to talk to you about making meaning of your data. You've set up your experiments, grown and observed your plants, and collected data. And now it's time to make sense of that data for yourselves and for others. The point of doing these experiments is to try to figure out what the effects are of genetically altering the Arabidopsis plants. We're trying to make a connection between the plant's genes and the plant's traits, and your experiments are part of that effort. So it's important to be as clear as possible when communicating your results back to your scientist partners. Here are some guidelines for organizing your results. Carefully review and organize the data you've collected. As you're organizing your data, reflect on your original research questions and ask yourself how your data might answer them. First of all, does the genetic alteration affect the growth and development of the mutant plants compared to the wild-type plants? And number two, does the genetic alteration affect the way the mutant plants respond to your experimental treatment compared to the wild-type plants? When you answer these questions, you will be making scientific claims. And we always back up scientific claims with evidence. As you review your data, ask yourselves which data best answers your research questions. Put your data into visual formats. For example, tables, graphs, photos, and other figures. Humans are visual creatures. This will help you analyze your results. If you haven't already done so, calculate the averages for the data you've collected. For each dependent variable, you should have one value for each pot of plants. If you've collected data more than once, use a line graph. The space between the lines will quickly convey the answers to your research questions. Is there a difference between the mutant and wild-type plants? A bar graph works great if you have a single data collection, such as the age of the plants when they started flowering. Put mutant and wild-type data on the same graph. You can put the control mutant and wild-type data on one graph and the experimental mutant and wild-type data on a separate graph if you'd like, but plotting the data from all four pots on a single graph is ideal. If there's a wide range in your raw data, then you can convey that in your graph with error bars. One of the simplest ways to see if the mutant plants responded differently to the experimental treatment than the wild type is to compare the percent difference in the control mutant and wild type data to the percent difference in the experimental mutant and wild type data. You simply divide the mutant data average by the wild type data average. Make sure you've clearly labeled everything: the units of your data, X and Y axes of graphs, and the title of your graph. Photos are best for presenting qualitative data such as color or shape, but if there's a clear difference in size, a photo can really help drive that point home. Write a paragraph describing the findings of each of the visual elements. Each visual element and its accompanying text should be able to stand alone and be easy to understand by the reader. And finally, arrange your visual elements, the tables, graphs, and figures, and their text descriptions in the best sequence possible to answer your experimental questions. Remember that you're trying to tell the story of what happened in your experiment. What did you learn about the Arabidopsis gene that you studied? So good luck, and contact me if you have any questions.